Today my webinar is going to be all about Peak's GlycanFinder version 2.5. So to start off, for anyone who may not know about what we do at Bioinformatics Solutions, we offer both software solutions for proteomics, antibody sequencing, and glycoproteomics. As well, we do have a lab here in Canada, and we do offer lab services for researchers who would like to send samples to us for us to analyze. But today's webinar is the primary focus will be on Peak's GlycanFinder version 2.5, which we released this version in early March. And we actually have a patch build that's coming out as well with some additional features. So just an outline of my talk, what I'm going to be going over today. First, just a general introduction to glycoproteomics. Then I'll go into the key features of GlycanFinder 2.5, focusing on our search algorithm and de novo sequencing strategy, the scoring that we do for confidence in the result, and then different fragmentation modes that we support in GlycanFinder. Then I'll focus a little bit more on some of the new features in GlycanFinder 2.5. This is including the release glycan workflow, and Peak's PRM for glycopeptides, so more targeted analyses. So just to start off, for a general introduction, protein glycosylation, it's one of the most common post-translational modifications in proteins, and it refers to the attaching of sugar molecules to amino acid side chains on proteins. These structures are quite diverse. They're built up of monosaccharide units that can take on these different branching structures. They are very important to many protein structures and biological processes, and as well, dysregulation is associated with various diseases. There's two different types of glycosylation when it comes to proteins. There's N-linked glycosylation, which the sugar is attached to an asparagine residue, and it nearly always comes in this motif where you have the N with the glycan attached, and then an N, XS, or S, T, or C motif afterwards. There's also O-linked glycosylation where the protein or the glycan is attached to a serine or a threonine residue on the amino acid. So glycoproteomics, it's a branch of general proteomics where with proteomics, we're wanting to know what proteins are present in the sample under a certain set of conditions. Glycoproteomics is an extension of that where we also want to understand the glycosylation modifications. Since they're so important to many biological processes, we want to include them in our studies of these proteins. So there's a few questions that glycoproteomics aims to answer. The first one is which glycoproteins are actually in the sample, so identifying peptide sequences and mapping them to proteins. What are the locations on the proteins of the glycan sites? This is sometimes referred to as macroheterogeneity because the sites are not always necessarily the same on every protein. And then there is also the question of what glycan structures and compositions exist at each site. This is referred to as microheterogeneity. And then we also want to ask sometimes, what are the relative changes in glycosylation between samples? Maybe is it different glycan structures across different species? Maybe it's different levels of glycosylation between healthy and disease states, for example. There are some challenges with studying glycoproteomics, though, and that partly comes through because of high glycan complexity. It can be very difficult to resolve kind of the different structures. Mass spectrometry is able to address these challenges because you have the mass spectrometry can differentiate the glycans with different masses. However, this mass spectrometry data is quite complex. And I just have here a couple of examples of the types of information that we get from glycoproteomics when we study through mass spectrometry. So I have here on the top a glycopeptide MS2. And we see that when we do the fragmentation, we get both peptide backbone fragmentation and glycan fragmentation. And so we can use the peptide fragmentation to actually sequence the peptide, and then the glycan fragmentation can be used to determine the structure and composition of the glycan. We can then, after we have these peptide sequences, map them to a database sequence, and then we can perform profiling to see, okay, if we look at all the different peptides that were mapped to this site, how many different glycan structures do we get? And then you can also do quantitative methods such as an area under the curve, like a label-free quantification or reporter labeling to determine the relative quantification of glycopeptides between different samples. And so these are kind of the type of data analysis that Peak's GlycanFinder aims to provide a solution for. Traditionally, the GlycanFinder software was specifically for glycoproteomics, so glycans that are attached to the peptide sequence. But we've recently moved more into Glycomics as well with our release glycan workflow that is new in this version. So just at a glance, some of the key features that we provide in GlycanFinder 2.5 are both N-linked and O-linked glycan profiling with structural resolution. So we report the glycans as structures and not just compositions. We have a database searching strategy combined with glycan de novo sequencing. This way you're able to identify glycans that may not be present in your database. We have glycopeptide quantification. We have a label-free option and a reporter labeling option with the ability to customize the reporter ion methods if the built-in one you would like isn't there. And then new in 2.5 is we have the targeted analysis, the glycopeptide PRM workflow, and a new release glycan workflow, which I'll be going into more detail a little bit later. And just a few other features, we have an intuitive database viewer where you can easily check the glycans that are in the database. You can easily edit, add new ones. We also have intuitive validation and reporting, and I'll get into how we do kind of the scoring and validation again in a little bit. And we also have vendor neutral data compatibility, so you don't need to convert your files to any sort of universal format prior to loading into GlycanFinder. You can just load the data in the raw format. So I'm going to go over the general search algorithm for our glycoproteomics workflows. It starts by taking an MS2, and the software will first identify whether or not it is a glycopeptide MS2 or just a peptide MS2. And it uses oxonium signature ions to figure this out. These are the signature ions that Peaks will consider when deciding whether it's a glycopeptide spectrum. You can take a look at these under the algorithm configurations in GlycanFinder. The next step, once it's been decided that it is indeed a glycopeptide spectrum, it will go through a peptide-based search. Some will refer to this as a peptide-first search. And this is where we attempt to match the peptide fragments to a protein in the database initially. And then once we have a peptide candidate, it will move on and try and match the glycan fragments to a glycan database. If this provides a good match, then it will be reported as a glycopeptide. If it is not, then it will go instead through a glycan first search, where instead of trying to look for a protein in the database that it matches to, it will just try and search the glycan database immediately. And this can be good when you maybe have a spectrum where the peptide backbone isn't as well fragmented. And if that's a good match, you'll have a glycopeptide reported. Once you have all your reported glycopeptides from the database search, FDR control will be applied at both the peptide match and the glycan match. For the peptide FDR, we use more of a traditional target decoy method with a decoy protein database. But with the glycan FDR, it's a little bit more complicated to do a target decoy calculation with a decoy database just because of the structure diversity. So instead, we actually use a decoy MS2 spectrum and we calculate the FDR that way. If it'll only be reported if both peptide and glycan FDRs are passed. And then anything that either doesn't pass this FDR, or if the spectrum didn't have a good match at all, it will undergo glycan de novo sequencing, and you'll have new glycans reported separately. And then this is just a view of our glycan database viewer. So you can edit, you can click on a structure, you can actually just click to branch out, draw your own new glycans. This is indicating here the sugars that we support in GlycanFinder. In addition to this, in the new patch build, we now have support for xylose as a sugar. It does require a little bit of configuring to get it to show up. So if that is something you're interested in, maybe you're looking at plant glycans, you can always reach out to us and we can help you figure out how to get that set up. But in the default configuration, these are the sugars that are available. In addition to, if you don't want to use the built-in database, if you don't want to create the database this way, you can load text files in GlycoCT format, and GlycanFinder can use those as well. This is showing our workflows for the discovery Glycoproteomics and Glycomics. So for Glycoproteomics, we have two options. We have glycan site profiling and then glycan sample profiling. For glycan site profiling, this is more geared towards multi-enzyme digests. You don't necessarily have to have multi-enzyme digests, but that was kind of the idea behind it. You provide it with the data. It will do a glycopeptide search, the de novo sequencing, and then you will get your report. And then this workflow is meant more to pin down the exact site of glycosylation. That's kind of how the multi-enzyme digest works. And then for the glycan sample profiling, you have your data, and then your glycopeptide search. It will also do a non-glycopeptide search, so you'll get non-glycopeptides reported as well if you choose this workflow. And then de novo sequencing. And then on the database search results, you'll have the option of either performing a label-free quantification or a reporter ion quantification before getting your result. Then for the released glycan sample profiling, which is new in GlycanFinder 2.5, you start with your data. It will undergo a glycan search, so there's no protein database involved because these were released glycans. And then you have the option of performing a reporter ion quantification. So these are the workflow options that we present in Peaks. I'll also go through the targeted workflow a little bit later. But first, I'm going to talk a little bit about the scoring that we do in GlycanFinder. So you'll get, for each peptide spectrum match, there will be an associated peptide score. And essentially you get that from a normalized sum of the supporting peptide fragment ions. You'll also get a glycan score that's also a normalized sum of the glycan fragment ions. And then you'll get a combined score. So it'll take into account both the peptide and the glycan score for an overall glycopeptide score to score the quality of this peptide spectrum match. In addition to that, we have the A score, which we call an ambiguity score. And that is telling you how confident are we that we assigned the glycan to the correct site. So there are some cases, for example, in this little picture here, there's an asparagine residue that has the glycan attached, but it also potentially could have been on this one. So we're assessing how good the peptide fragmentation is to be able to differentiate these sites. And also we have an S score, which stands for a structural score. And that is evaluating if there's multiple glycan structural candidates, what evidence is there in the glycan fragments that it is one structure over another. So I'll go into a little bit more detail with the A score and S score. So the A score can be interpreted as a -10 log p-value, where the p-value is the probability that the modification occurs at any possible position other than the reported position. So essentially the probability that we made the wrong assignment. So a higher A score is better than a lower A score. And I have a few examples of some scenarios that you might encounter with this. The top one is showing an A score example of 1,000. And you can see in this example, there is quite nice fragmentation along the peptide backbone, but there is also only one possible site for this N-glycan. That makes it really confident due to indirect localization. So we were able to exclude all sites based on there only being a single possible residue. So often in those cases, you will see extremely high A scores. In the second scenario, we don't have that ability to exclude. There are multiple asparagenes in the sequence. As well, there's some poor fragmentation around the one that was actually assigned. And so this one has a little bit of a lower A score. There's still some confidence because you do get some fragmentation between the two possible sites, but it's not quite as confident as the first result. And then this last one is showing an example where you would have a really poor A score where you have 3 possible sites in a row from each other and then really poor fragmentation along the peptide. And in this case, sometimes you'll see a score of 0. So this is overall showing the confidence of the glycan site assignment. And now taking a closer look at the S score. So this is reported as a percentage, and it's the confidence of the top glycopeptide compared to other candidates for the spectrum. And it's based on the number of matched glycan Y ions that are counted. So in the actual software, you will see a mirror plot with the top most confident one on the top, and then on the bottom, the second most confident one. And you can actually switch, look at all candidates, and look at the spectra for every single candidate if you would like. But the actual S score, you're taking the glycan structure with the most Y ions. You're taking the number of supporting Y ions and subtracting the second most Y ion count, and then dividing it by the first most Y ion count. And so what that gives you is a percentage, and 100% S score would mean that there is only one candidate, there aren't any others, whereas an S score of 0, for example, would mean you have exactly the same number of fragment ions supporting that structure. So it's telling you how confident are we in the structure assignment, and how confident can you be that there are no other possible candidates based on your fragmentation. I also want to talk about fragmentation because a key challenge in glycoproteomics is obtaining that adequate fragmentation both along the peptide backbone and on the actual glycan modification. And in Peak's GlycanFinder, you can get the best score when you have good fragmentation along both peptide and glycan. And so oftentimes the choice of fragmentation mode can make quite a big difference into how good this fragmentation is. And a lot of researchers employ different strategies. A lot of mass spec instrument vendors have different fragmentation types. Some of the general ones that are used for glycoproteomics is CID and HCD methods. These are often known to provide very good glycan fragments. So we have this spectra here is CID showing good peptide or glycan fragmentation. And then this one up here is also, or is an HCD, and you're seeing good glycan fragmentation, but not necessarily as good peptide fragmentation. Whereas electron methods such as ETD and EAD often provide a better backbone fragmentation, but maybe not as good glycan resolution. So sometimes in order to get both, hybrid fragmentation modes might be used, such as a hybrid between EAD and CID. Sometimes ETHCD is used, or a combination of ETHCD and HCD. Also, stepped collision energies are used as well. And so the point I'm trying to make here is that in GlycanFinder, we're able to handle many different types of fragmentation modes, regardless of the instrument vendor. So the peptide score will consider different ions that are known to be produced by certain fragmentation types. When you load your sample, you'll be able to choose your fragmentation types between CID, HCD, ETD, mixed, ETHCD, and EAD. And I've just shown some examples of how certain spectra might look as an example in Peak's GlycanFinder. So we can see in this CID example, you have some nice glycan fragmentation and a decent peptide fragmentation, but the B and Y ions are a little bit smaller, less intense, and that's very typical of a CID or an HCD spectrum. And then we have an EAD where you see the C and Z ions are a little bit more intense than the glycopeptide one or the glycan ones. But then the actual glycan fragments aren't really giving you that much structural resolution. And then a combination of EAD and CID, this hybrid fragmentation mode, you're able to get good peptide backbone fragmentation, a mix of B, Y, and there's a C ion there you can see, and then others that are just smaller that aren't really obviously visible to us without zooming in. But then you also get decent glycan fragmentation. Over here is showing a stepped HCD that these researchers did, where again you see nice glycan fragmentation, not as nice peptide fragmentation. ETHCD, you see kind of the opposite, where it's better peptide fragmentation. And then this is a mix of stepped collision energy for HCD, and then ETHCD is giving you kind of better results for both. So this is just kind of demonstrating that in Peak's GlycanFinder, you're able to make use of all these different fragmentation types, regardless of the strategy that you use. So now I'm going to move on to discuss some of our new feature highlights. First, I'll be going over the release glycan analysis workflow with the reporter ion quantification. So just as an overview, free glycans, which aren't attached to proteins, are how we describe release glycans. Typically in a glycoproteomics experiment, they may have been initially attached to a protein, but they may have been removed during the experimental setup. This kind of analysis can provide a nice complement to glycoproteomics because even though you are losing that protein information, that site-specific information, you're able to almost get a deeper look into the glycan structure itself, mainly because it's a simpler fragmentation for MS2 analysis. You only have to worry about that glycan structure. Some of the highlights of our Peak's GlycanFinder release glycan workflow, and some of these features are from the recent patch build. So if you are a current user of 2.5 and you don't know about some of these features, some of them are a little bit newer, but we do support glycan modifications. In the patch build, we've added support for permethylation, cross-ring fragmentation analysis, adduct consolidation for quantification, and then reporter ion quantification methods. So for glycan modifications, we do have these built-in modifications that you get automatically, and you can set fixed and variable modifications for the sugars. You can also, in addition, customize your modifications. So if you don't see the one you want to search for in the list, you're able to create your own modification. And again, we've added support for permethylation. So if you do have permethylated release glycan data, you can just check the box and GlycanFinder will do the search, assuming that you've permethylated your sample. We also have the ability now to analyze cross-ring fragmentation. So of course, when we're talking about proteomics, we have these ABC, XYZ ions that can come about as different parts of the peptide are fragmented. And for glycans, there is a similar naming convention, just with capital letters instead of small letters. In the glycoproteomics workflows in GlycanFinder, we're mainly focusing on the Y ions, with the rest of the sugar being attached to the peptide for the most part. However, for the release glycan workflows, we take into account all of these ions, including the A and X ions that can indicate that actually can cleave across the sugars themselves. So instead of between the monosaccharides, they cleave across the ring, which is why they're called cross-ring fragmentation. So if Peaks finds one of these ions, it will annotate it as, for example, an X 2,4. You can see an example in this spectrum, and that will indicate where in the ring it has cleaved. So that can be helpful for getting maybe some linkage information. That's a bit of a deeper look into the structure than for the glycoproteomics. Another feature we have is adduct consolidation. So sometimes with released glycans, you can be a lot more prone to adduct formation than you are with glycoproteomics. This is an example here showing 3 different glycan features. They all actually lead to the same glycan identification. However, they all have different adducts. So this first one here has no adduct. This one has a sodium adduct. And then this last one has an ammonium plus a sodium adduct. So they're all different M over Z values because they're all different masses. They're all slightly different retention times because they're different properties. However, they all belong to the same glycan. So what Peak's GlycanFinder will do is it will report these all under the same glycan. And that way, if you go to do a quantification, it will have an accurate representation of that glycan. And here's just an example of how it will look in a search result. So you can see here, this is the same glycan that was reported. I just used this feature selection breadcrumb to choose 2 different features. This one at the top here has a sodium adduct, and then this bottom one here does not have a sodium adduct, but you can see they're both reported under the same glycan entry in the result. We also report a glycan network as part of the summary page of the released glycan workflow. And this is where you can see different connections between the glycan compositions. So what this does is it links glycans that differ by 1 monosaccharide. So you can kind of visualize that you might have a certain core of a glycan, and you can visualize maybe which sugars are branching off of that core structure, and it can help infer different clusters of glycan synthesis. Now to talk about reporter ion quantification a little bit. So I'm going to use some published data to illustrate how Peak's GlycanFinder handles reporter ion quantification of released glycans. So the Lee lab developed these sugar reagents. So sugar in this case stands for isobaric multiplex reagents for carbonyl containing compound. And they're essentially a reporter tag for glycans. They work very similar way to reporter ions that are for proteomics, such as TMT reagents, where they have a reporter region, a mass balancer region, and then a reactive group. And then each label or each tag within the group has a different reporter and mass balancer, but the overall mass is the same. Each different condition will be labeled with a different tag and then mixed together. This is where the reactive group comes in. In this case, this will react with the reducing end of the sugar. And then what happens is when you run on the LCMS, so the same glycans will be indistinguishable from each other, and so they'll be treated the same in the LC and in the MS1. And then at the MS2 level, when the fragmentation occurs, you'll get unique fragmentation from this reporter region of the reporter ion, and you can use that for relative quantification between the groups. So again, very similar to how it works in proteomics. This sugar reagent is actually built in automatically to GlycanFinder, but again, you can customize your own reporter ion quantification if you have your own reagents. So this is just an example. I ran some data in GlycanFinder where, again, developed by this group, they had these 12-plex sugar labels. I've just shown a picture so you can visualize the reagents that were used. In this experiment, ECC1 cells were cultured and then divided into 4 groups. And then 2 of the groups were the control. They were treated with DMSO. And then the other 2 groups were the treatment groups, and they were treated with an antimicrobial medication, atovaquone. And then they were either treated the groups for 48 hours or 72 hours with the respective treatment. So that's 4 different groups in total. And then each groups were treated with different sugar labels with 3 replicates. So essentially 3 different labels per group. And then this is showing in Peak's GlycanFinder the reporter ion quantification result. So you can see here the actual glycan identity is listed, the sample profile heat map, the group profile heat map, and then intensities across the different groups. You can also see the MS2 with the evidence for that particular glycan structure, the MS1 feature, and down here you can see the reporter ion groups. So you can see there's multiple here. Some of them are very close together, so it's a little harder to distinguish visually, but it will show you all the reporter ions here. And what we see from this group is overall the glycan levels decreased 48 hours after the treatment compared to the control group. So that's these blue bars here. These are the top 7 essentially glycan structures from the reporter ion quantification result. And then it was also found that after 72 hours, they returned to near levels of control. So that's the orange group here. And we noticed that the difference is pretty subtle and the reproducibility wasn't amazing, but that actually was a very similar result to what they found in the publication. So this is an example showing how GlycanFinder can be used for this type of application for reporter ion quantification of different treatment groups. The last thing I'm going to be going over today is our new glycan PRM for targeted proteomics, or for targeted glycoproteomics. So everything we've talked about up until now has been more discovery oriented, where you're taking your sample, you're searching it against a database, or you're doing de novo sequencing, and then you're finding out what is in that sample. Whereas with targeted, you already know specifically what you're searching for. And so I wanted to test our GlycanFinder PRM workflow again against some published data where they had some structures to verify by PRM. So in this paper, they were trying to identify possible glycopeptide gastric cancer biomarkers. And they did this by initially searching large discovery and validation cohorts to come up with certain glycopeptides that presented significantly more in the cancer groups than in the healthy control groups. They came up with these 4 structures here. And then they also verified these structures by PRM. So I kind of wanted to recreate that in GlycanFinder 2.5 to see how the PRM workflow could work with this type of application. To start, I did an initial glycopeptide search before the PRM. This is actually a good thing to do before running the PRM workflow because it's an easy way. When you have a search result, you can export the glycan transition ion TSV, which will be required for the PRM workflow. In this example, I ran the sample profiling workflow, but you can also run the site profiling workflow and also get a transition list from the result. So I wanted to do the initial glycopeptide search just to double check that I was getting the same identifications on those peptides, that I was actually seeing them in the sample before I used the PRM workflow. And I've just shown here an example of one of the sites identified in the paper, just to show kind of how it would look in GlycanFinder 2.5. So this was for site N91 of this inter-alpha trypsin inhibitor heavy chain H3. You can see here just in the protein coverage result, if you were to click on this asparagine with these glycans on it, it will bring up this glycan profiling pie chart where, based on the areas of the supporting peptides, it will tell you the relative abundance of that particular glycan identity compared to other glycan identities. And we can see in this case that actually the most abundant one was indeed the biomarker that was identified by the paper. But we can also see what other glycans were present at that time too. And then I looked at the site profiling. So within Peak's GlycanFinder, this would show separately by sample. However, just to make things easier, because there was about 100 samples in there, I averaged the healthy controls, which is the blue, and then the cancer group, which is the orange. And we can see that for the biomarker that they eventually identified in the paper, it is indeed quite a bit stronger in the cancer group. And you can see it's not as big of a difference for the other glycans. I just plotted the top 4 highest area glycans for this particular site. I also want to highlight, before I dive into the PRM workflow a little bit, one feature that we have that has to do with this glycan profiling pie chart is the charge lookup feature. So what charge lookup is, it's essentially like a match between runs, if you're familiar at all with that concept in the context of label-free quantification. So in charge lookup, you might have one feature that a certain charge state that has a good identification. Maybe you have another feature that based on M over Z and charge, you can tell it's the same peptide, but it for whatever reason didn't get a good quality MS2. GlycanFinder will do a charge lookup, and it will actually apply kind of that identification to the missing value, and it will include that area in this glycan profiling pie chart. So that kind of gives you more of a complete result. You're not missing out on anything that maybe you missed an identification with. And then this is an example too, if you were to look at the glycan profiling of this protein, you can look by site. In this chart, it's going to show you separately by each sample the abundance of the different glycan identities. So again, it's just by separate samples. So it's the same thing I showed you before, it's just not averaged. And this is a different example. So now moving on to the PRM. When you're loading data as PRM, you'll want to select under acquisition mode PRM instead of DDA. And then you will pick the Peaks PRM workflow. You'll need to also provide a transition list. So, excuse me, you can either export the TSV file provided in your database search result, or what you can do is you can just create it by hand if you want, but the information required are peptide sequence, the glycan sequence in site, adduct information if applicable, the M over Z charge, and retention time window. So that's what you're providing instead of a database. This is a very specific search. Again, you have your targets already. You know what you're doing. In this case, I just provided the same targets that they searched for in the publication. You have a couple of options as well when doing the PRM workflow. You can choose the precursor and fragment, peptide and glycan fragment separately, the error tolerances that you want to use. You can pick your base sample. So for ratio calculations, this is essentially picking what the denominator is going to be. And then you'll add your transition list. And then we use the fragment ion area under the curve for quantification. So that is similar to how we do it in our LFQ DIA workflow in Peak Studio, if anybody is familiar with that type of workflow. By default, it will use the top 3 fragment ions for quantification. However, this can be configured to use more or less fragment ions, so you can customize it in that way. And then this is the PRM result page. So you have the formula, glycan, the significance. So that's comparing within the sample versus across the groups. How similar is the result? M over Z charge, the glycopeptide score, and then the areas for each groups and the maximum ratio. You'll also see the actual supporting MS2 spectrum with the supporting fragment ions for the peptide and the glycan. And then you'll be able to see the precursor profiles and the fragment ion profiles. And you can check or uncheck different different samples to see different precursor profiles for different ones. And then comparing back to what they got in the publication, so Peak's GlycanFinder was found to identify the same structures as the publication. It did identify one additional structure here that was for this site that was not considered in the publication. But again, you can look in the actual result and you can take a look at the supporting fragment ions to see what the evidence is for each structure assignment. But overall, we were able to confirm that we could get the same result in the PRM analysis. And then actually looking at the areas, so I just made up a few figures from the results in Peak's GlycanFinder in a similar style to what they did in the publication. And we observed that it was generally similar ratios observed as in the publication, where the markers were slightly higher in the gastric cancer patients compared to the controls. And showing that Peak's PRM for the glycopeptides is a good method to get an accurate result and also save time on the analysis because there's no need to do anything manually. It does the quantification for you. So to conclude overall, hopefully I've been able to show you today that Peak's GlycanFinder can provide a comprehensive solution for Glycomics and Glycoproteomics. Combining the peptide first and glycan first searches with the de novo sequencing really allows for sensitivity across your sample, not leaving out any spectrum, maybe if the peptide fragmentation isn't so good. There's multiple scoring parameters for the best result confidence. And as well, hopefully our new features, such as the release glycan analysis or the PRM, can help researchers to use this software for some more additional applications. So with that, I would like to thank everybody for listening today. If you wish to get in touch with us at all, you can reach out to technical support. The easiest way is emailing support@bioinfor.com. We're happy to answer any questions about the software, about how anything works, whether it's for GlycanFinder or any of our other software options. You can also, if you're interested in getting a quote, a one-on-one overview for any of our software, or if you're interested in setting up a trial for any of our softwares, you can reach out to sales@bioinfor.com or call the phone number. And as well, at the same email and phone number is our lab services sales if you're interested in our lab services at all. With that, that is my talk for today. I'd be happy at this point to answer any questions if anybody has any of them. You can either unmute yourself and ask verbally, or you can type a question in the chat.